Good evening, and a very warm welcome to this webinar on preparing for the future, harnessing the power of genomics and vaccinology to tackle pandemic threats, in which we will delve into the intersection of genomics and vaccinology and how these have revolutionized vaccine development and on the lessons learned during the COVID-19 pandemic. We have two excellent speakers with us, and I will introduce them shortly. Before I go on and do that, I want to just give you the announcement that the annual meeting for the annual APAM meeting is going to be on the 20th and 21st of April of this year, and early bird registration finishes at midnight tonight. So just to remind you to register for that. And now an introduction to this webinar. So in the response to the SARS-CoV-2 pandemic, the use of mRNA and development of vaccines has been really crucial. The rapid sequencing of the virus has allowed us to have a better understanding of its genetic makeup, leading to the development of very highly effective vaccines. And real-time monitoring of the virus's evolution through next-generation sequencing techniques has allowed for the rapid adaptation of vaccines to new variants. And we will explore the impact of these advances on the current pandemic response, and then what it means for the future of vaccine development for this and other viruses. I think that we'll have a great opportunity to gain insights into the latest breakthroughs in genomics and vaccinology. Just to introduce myself, my name is Emma Thompson. I'm Professor of Clinical Infectious Diseases at the MRC University of Glasgow Centre for Virus Research. And I'm joined by my co-host, Julian Knight, who's Professor of Genomic Medicine at the University of Oxford, who you probably mostly know well, and who is also an expert in genetic variation and how that can impact genes critical to mounting appropriate immune responses and susceptibility to infectious, inflammatory and autoimmune diseases. Now, moving on, I'm going to introduce our first speaker, Professor Sharon Peacock, CBE, who is a British microbiologist and is Professor of Public Health and Microbiology at the Department of Medicine, University of Cambridge. She's very well known for her use of microbial whole genome sequencing for bacterial infections, but in 2020 she became Executive Director and Chair of the COVID-19 Genomics Consortium, or COG-UK. And she oversaw the development of a network that catapulted the UK to prominence as a leading authority on the generation and use of SARS-CoV-2 genomes as part of the pandemic response. The emergence of variants and the need to detect and track these represented a major shift in thinking and practice for disease control and vaccine effectiveness. And perhaps I can now welcome Sharon to give us an introductory talk. We will then move on to Tess Lam, who I'll introduce before her talk. Sharon, if you'd like to.
Yeah, thank you so much, Emma, and thank you so much for inviting me to talk today. It's a great privilege and pleasure to be here today. Okay, so let me just put my slides up. The moment of truth. Can you see my slides okay? Brilliant, okay. Well, the title of my talk is Lessons Learned from Sequencing During the Pandemic. And I've split it down the middle. I'm going to talk about what we learned about the virus, but I'm also going to talk about how we learned to do rapid sequencing very well and for the benefit of the pandemic response. And so the lessons learned really are kind of, I suppose, a scientific lessons learned, but also a kind of a reflection on how we managed to stand up such a large consortium in such a short space of time and deliver a large number of genomes. Okay, so we were not the first person to sequence the virus, not by a long shot, actually. So there was a tweet by Eddie Holmes on the 11th of January 2020 saying that there was initial genome sequencing of coronavirus associated with the Wuhan outbreak. And that was then reported in full in January. And that information was absolutely key to vaccine development, because we then understood that this was a coronavirus, that it had a spike protein gene, etc. And you may well hear more about that from Tess, but that's That was the vital first clue, what we were dealing with here, what the cause of the infection was, and how one might respond to it in terms of vaccination development. But actually what the UK did do was it was amongst the first to develop a capability to generate SARS-CoV-2 genomes at scale and use this for public health, because in the past, we've tended to use sequencing of pathogens quite reactively and often used to look back to solve kind of a query or a question. What we did here was we used it to guide the pandemic response. And we really showed beyond any doubt, I think, that sequencing generates actionable information that supports an effective response through vaccine development and redevelopment, understanding transmission of the virus between people and across the world. And so I'm going to just talk a little bit now about the basis of what we were really looking for. So what we were really looking for was to track viral mutations. And so mutations in the genome are the basis of most evolutionary change. And RNA viruses have high mutation rates, actually, up to about 1 million times higher than the human genome. The issue is that changes in the virus have the potential to allow them to adapt to their environment and continue to infect humans. Now, this shows a schematic of the viral genome of SARS-CoV-2. It's around 29,000 nucleotides long. And you can see in the pink the gene that codes for the spike protein. Now, the spike is important because the spike is studded onto the outside of the virus, and that's where the word corona comes from. It forms a crown. And that spike is really key because it's like a lock and key. It's the thing that recognizes the human receptors or the way into our cells so that it can replicate. And some of the work that Emma's done on how the virus has got into cells and how that's changed over time has been a really elegant description of virology during the pandemic that really helped us to understand disease severity and other aspects. So this spike protein is really key, and it's the place where our immune response is targeted to, because if we can get an antibody blocking the spike, then we can prevent the virus from attaching and entering. So SARS-CoV-2 mutations, they happen probably about twice a month in the generation of a virus. And there are 3 outcomes. Most mutations that can occur, they're completely random. Most mutations, they don't affect the virus behavior at all. Some mutations will disadvantage the virus, actually, and though the progeny of that virus will not be observed in the population or will dwindle. What we really want to know about is the mutations that cause an advantage to the virus in terms of its fitness properties compared to everything else that's circulating. So this is a process of natural selection where the fittest will win out. Now, having trained as a clinician, and the ABC of resuscitation was drummed into me: airways, breathing, and circulation. The way I think about variant assessment for COVID-19 is to think, is the virus more transmissible? Is it likely to evade the immune response, either because we have had natural infection before, we have antibodies, or we've had a vaccination? Or is this going to cause more or actually less severe disease? So this is the variant assessment. But however, spotting important variants was very difficult and still is, actually. So spotting which mutations are associated with the change in biological behavior. If we go back to September, October, November last year, we had very little understanding of what that actually meant. And over time, we've accumulated a catalogue of mutations that we believe are associated with particular characteristics. But initially, it was a very steep learning curve for us, and spotting important variants in amongst the many thousands of variants that were circulating was challenging. And this is a schema that was developed by public health agencies, including PHE and US CDC, where really what you're doing is you're horizon scanning for the behavior of the virus in the community. Is it reinfecting people who should be immune? Is it spreading more quickly than you would expect normally? So horizon scanning really is looking at the biology of the virus in the community. And if there's a signal, such as it seems to be spreading more, then it becomes a variant of interest. And at that point, the investigation really begins in earnest to watch what that variant is doing. Now, we know there's been a series of variants of concern, and these are ones that spread more rapidly and or evade our immune response. And then very fortunately, we haven't seen the top of that, ladder, which is a variant of high consequence, which would be a virus, for example, that caused much more severe disease, completely avoided our immune response, etc. So this is one of my favorite pictures of all time, actually, and it's an image from MicroReact, developed in Oxford by David Anderson and his colleagues to look at the variants in the UK circulating over time. Now, if we look at these, this is a timeline from March 2020, and it actually ends quite shortly after Delta has really become dominant in July 2021. If you look at wave 1, and each colour is a variant, not a variant of concern, just a variant, a specific combination of mutations in the virus. You'll see in wave 1, there was a huge number of variants that were circulating, and they were all, I guess, competing against each other. Nothing was really escaping. Then at the beginning of wave 2, we saw something called B.1.177 in the red, and it expanded to create, it was causing around 30 or 40% of all cases in the UK. And detailed investigation demonstrated actually that this wasn't due to the virus, it was due to holiday behaviour. So people were going to Europe, they were acquiring this in Europe and introducing it into the country, and then it would spread. And so things that expand aren't always to do with the biological behaviour of the virus. But when the Alpha variant emerged, which you'll start to see really starting to begin to emerge in around middle of November onwards, what we then realised was that this virus was spreading more than it should do, considering we had already gone into lockdown. And what you'll see here is this pattern of Alpha completely wiping the board clean, and it went on to sweep across the rest of the world. And then Delta came along and swept that away. And then, of course, there's been Omicron since. So what this again shows really is how we learnt about the virus. We fully understood the rules that we were playing with in terms of, you know, Darwin's natural selection, but it was really only in December and onwards that we saw these sweeps of variants that were clearly fitter than other variants that were circulating. And of course, we know that Alpha variant emerged, was detected in December. And we can talk about this if you're interested, but it had acquired multiple mutations. There's been quite a lot of discussion about what appeared to be kind of supercharged evolution in this variant, and it was more transmissible, about 50% more transmissible. Now, we called this the Grinch variant because actually it did steal Christmas from everybody. We had to go back into lockdown, but COG-UK were called back into work over Christmas to sequence as fast as we could to understand this virus better. And since then, we've had waves of variants. In fact, this doesn't even come up to the minute, but you'll see the way we've had sweeps of variants go through our population because they have an advantage in transmissibility and or vaccine escape. This is one of my favorite images of the relative fitness of SARS-CoV-2 variants over time. It looks quite complicated, but it isn't that different, isn't that complex. It was published in Science in 2022. And on the y-axis, you've got the fold increase in relative fitness of a variant compared to the original virus, which we call the Wuhan lineage. And what you'll then see on the x-axis is the date of lineage emergence. The size of the bubble is the number of people who were diagnosed, it's cumulative case count effectively. And what is extraordinary about this is that the Omicron is, as you can see at the top right-hand corner, much fitter than other variants, but fortunately causes less severe disease. And again, the work that Emma did was key to understanding the basics, some of the basis for that. So what's the future scenarios for the SARS-CoV-2 virus? Well, there are 4 possibilities. One is that the virus becomes extinct. It just dies out pretty much through minimal control, or the virus isn't really very fit. Global eradication through vaccination, for example, with smallpox, we achieved that. Elimination through zero COVID policy, well, we know that that was attempted but didn't succeed in the end. And endemicity, that's where we are at the moment. We're going to live with this virus for the foreseeable future. So just as I come to a close, I'm actually going to think about my personal top 8 lessons, I don't know why 8, it's just I added them up this morning after having done the slides. And I want to run through how we can think as scientists, not just about the science, but the way we behave when something like this happens. Now, the first thing that actually helped us to respond very quickly to sequencing was how well prepared we were for the future. We had deep roots, so we had an academic engagement on pathogen genomics for well over a decade. We had equipment, consumables, know-how across the UK. And the 100,000 Genomes project was largely focused on human genomes announced by David Cameron, but we actually, as a recommendation from that, said that sequencing of TB, HIV, and hepatitis C should advance at pace for the benefit of patients. And so the expertise of applying genomics was really beginning to gather pace. We also had real-world experience and practice with outbreaks such as MERS, Ebola, and Zika, and the CMO was behind us in publishing a chapter in her volume 2 2016 Generation Genome, and that highlighted the really many benefits for sequencing of bacteria and viruses. Science investment was really important, so we had the MRC UKRI funded CLIMB, which is a cloud-based infrastructure for microbial bioinformatics. That's where all the analysis goes. And the methodology network for doing sequencing called Arctic was really thriving. And of course, we have the kind of stupendous Wellcome Sanger Institute, a mighty institution that can do very high volume sequencing. So unlike for other areas of our response, we were well prepared for this. We also acted at pace, and we were funded at pace. And so I'm not going to go into detail in this timeline, but it's a timeline of March 2020. And you can see on the top the number of genomes we had sequenced during March, and on the bottom, the number of cases that were diagnosed during March in red, and underneath the number of genomes we had generated. So what we decided to do was a group of scientists got together on the 11th of March at the Wellcome Trust to decide a blueprint for how we were going to be sequencing this virus. We wrote within a couple of days a proposal that we submitted to Patrick Vallance and Chris Whitty, and they agreed to fund that by the end of the month. But by the middle of March, we were already getting teams underway to think about how we collect samples, how we sequence, how we link the genomes to the metadata, how we report right, etc. And something called CLIMB COVID was created, which is our cloud infrastructure. So acting at pace was really important. Teamwork was also really crucial. So this consortium had 21 partners. The 4 public health agencies of the United Kingdom were really the kind of the levers for implementing what we found, and 16 academic institutions across the country. So you can look to see if your institution is there, and also the Wellcome Sanger Institute. And we had a consortium agreement that was a wraparound, a legal framework that allowed us to work together. But teamwork was key, as well as a focus on impact over process. Another lesson I really learned was I had to work in an imperfect system, and so did everybody else. So people said that it was like rebuilding the plane whilst trying to fly it, and we all had a piece of it. So we were desperately trying to get through the day, and the whole system was constantly changing. So a new lighthouse lab would open, for example, we'd have to get their samples or another diagnosis. There was lots of things changing the whole time. So I didn't find it particularly comfortable as a scientist who likes to be, think about kind of the perfections in life, but this was an imperfect system that we were working in. And it was also very chaotic at times. We also innovated, but we didn't have a central control and command. We devolved the innovation to teams, and we had devolved leadership. So we had different teams being led on logistics, methods development, data linkage, and analysis tools. And to devolve that was really important for us. We also shared what we created. So the work of COG members was adopted worldwide. We had sequencing methods, the Pango lineage, informatics tools, and we released all of our genome data immediately that we had it to the world. I think this is my 7th lesson. We got someone else to mark our homework at the end of the day because we wanted to know where we'd done well and where we had done less well. And so RAND Europe was asked to do a full evaluation of our performance. It's on the internet should you want to go and see that. And so important to finish with, actually, is that it's the people that really matter. And one of the things that we did was there were about 600 people in our consortium, mostly volunteers. Our money went on sequencing rather than on salaries. And so the people really made this work, but we shared the publication dividend by ensuring that we had a COG-UK title that went into every paper that used our information. In that way, we could honour the people that have helped us make this consortium a spectacular success. So a big thank you to all of those people and their families, actually, particularly over Christmas 2020. So I'd like to thank you for listening, and it's been a pleasure to talk about this subject to you today. Thank you.